We left off on the last lecture with an introduction to plant stems and a focus on vascular tissue. As mentioned previously, the vascular tissues shown here on the slide are part of a continuous pathway that extends throughout the plant's 4 organs. The vascular tissue can be divided up into the xylem and the phloem. The xylem is found towards the interior of the plant and is the site for water transport. The phloem is found towards the outer edge of the plant and is responsible for the transport of the products of photosynthesis. Additionally, both the xylem and the phloem also function to transport important signaling molecules, like hormones and dissolved nutrients. Water travels with ease through the xylem because of the structure of the water-transporting cells within this tissue. As they develop, these cells become greatly elongated. When they complete their growth, they secrete and build a thick secondary cell wall that contains lignin, a chemical compound that increases mechanical strength of the wall but also impedes the movement of water through it. Finally, the nucleus and the cytoplasm are lost, leaving behind only the cell walls. The thick walls form a hollow conduit through which water can flow. Water enters and exits the xylem conduits through pits. Pits contain only the thin, water-permeable primary cell wall that surrounded each cell as it grew. Pits allow the passage of water, but not air, from one conduit to another. There are 2 types of cells or conduits found in the xylem. The first are tracheids. These are single cells which slightly overlap with their neighboring tracheid cells. Water enters a tracheid through a pit, travels upwards through that tracheid cell or conduit, and then enters the next adjacent, partially overlapping tracheid to continue on its upward trajectory. This means that as water is transported via tracheids, it must pass through primary cell walls as it moves from cell to cell on its journey. Tracheids are the major conduit in lycophytes, ferns, and horsetails, and in most gymnosperms. The second type of conduit found in the xylem are vessels, which are long multicellular conduits made of individual vessel elements. Vessels can be much wider and longer than tracheids, and they are found in almost all angiosperms. As water is transported in vessels, it must also initially enter through pits. In contrast to tracheids, however, once the water is inside the vessel, little or nothing blocks the flow of water from one vessel element to the next. During development, the ends of the vessel elements are digested so that the column of vessel elements forms a hollow tube of cells that make up the vessel. At the end of a vessel, the water must flow through its pits if it is to enter an adjacent vessel, and thereby continue its journey from the soil to the leaves. Because vessels can be both longer and wider than tracheids, plants that have these conduits can transport water at a greater rate. Additionally, since the water in plants with vessels does not need to move across pits as often, it faces less resistance during transport. Bulk flow, like that in the vascular system of plants, is propelled through differences in pressure. In the case of water transport, transpiration drives the difference in water pressure. When stomata are open, water evaporates from the walls of the cells lining the intercellular spaces of leaves. The partial dehydration of the cell walls creates a force that pulls water towards the sites of evaporation, starting with the adjacent cells. Once generated, this force is transmitted through the xylem, beginning in the leaf veins, and then down through the stem and through to the roots in the soil. Transpiration lowers the pressure of water in the xylem and the leaves, which then causes the water to flow from the soil to the leaves, thus creating an evaporative pump. Water can be pulled through the xylem because of the strong hydrogen bonds that form between water molecules. This mechanism of water transport only works if there is a continuous column of water in the xylem that extends from the roots to the leaves. The forces that pull water from the soil in plants must be able to lift water against gravity and pull water from the soil, even when it is dry. These forces must also be greater than the physical resistance associated with moving water through the plant itself. There are several different processes that impact the movement of water through plants. The parameter that combines these factors is called water potential. Water potential accounts for factors such as pressure, osmosis, and gravity. Water will move from areas of higher water potential, such as the roots, to areas of lower water potential, like the leaves. Negative pressures pulling water up through the plant was first proposed in 1895, but at the time there was not a way to measure the magnitude of these pressures. In 1912, the German physiologist Otto Renner hypothesized that the suction produced by leaves are stronger than those that could be generated even with a vacuum pump. To test this, Renner set up a neat experiment where he cut the tip off of a branch from a transpiring plant, placed the cut into an apparatus which allowed him to measure the flow of water being sucked into the branch via transpiration. He then cut off about 10 centimeters of the branch, attached a vacuum pump to the branch, and measured the flow rate generated by the pump through the branch. Renner found that the flow rates generated by transpiring plants were 2 to 9 times greater than the flow rates generated by the vacuum pump. The maximum suction that a vacuum pump can generate can lift water about 10 meters. Plants can pull water up to heights of more than 100 meters. Transport of carbohydrates takes place in the phloem. Phloems consist of multicellular sieve tubes composed of highly modified cells called sieve elements that are connected end-to-end into sieve tubes. Unlike the xylem, the phloem is comprised of living cells at maturity, though during development, much of the intracellular structure of sieve elements is lost. At maturity, their cytoplasm contains only smooth endoplasmic reticulum and a small number of organelles, including mitochondria. The sieve elements still maintain a plasma membrane, however, though they do no longer possess a nucleus or a vacuole. Cellular functions such as protein synthesis are carried out by an adjacent cell called a companion cell, to which the sieve element is connected by numerous plasmodesmata. Sieve elements themselves are linked by sieve plates, which are modified inwalls with large pores. As you look down through a sieve element, the sieve plates look like nets, as you can see in the photo in the bottom left. The plasma membrane of an adjacent sieve element runs through and lines the pores in the sieve plates, so that the cytoplasm of one sieve element is continuous with the next. This means that even though there are multiple sieve element cells stacked end to end, they can be considered to form a single cytoplasm-filled sieve tube. Phloem sap is the sugar-rich solution that flows through both the lumen of the sieve tubes and the sieve plate pores. Phloem transports carbohydrates, amino acids, inorganic forms of nitrogen, ions, hormones, protein signals, and RNA. The phloem transports its molecular cargo from sources to sinks. In other words, the phloem transports carbohydrates from areas where they are made or stored to areas where they are needed. Sources can be the leaves, where the sugars are produced via photosynthesis, or storage regions, like tubers, where the carbohydrates are kept until needed. Sinks are regions of the plants that need sugars to fuel growth and respiration. This can include the roots, young leaves, and developing fruits. Storage regions can also function as sinks while the sugars are transported there for use later. Transport through the phloem can either be up or down the plant depending on where the source and sink are relative to each other. Transport in the xylem depended on an evaporative pump. Transport through the phloem uses an osmotic pump to drive the movement of sugars. This pump results from the differences in sugar concentration between sources and sinks. The concentration of sugars in the sources is relatively higher than the concentration at the sinks. This higher concentration pulls water into the phloem via osmosis at the sources, increasing the turgor pressure there. At the sink end of the phloem, sugars are removed from the phloem. This causes water to leave the phloem, and the turgor pressure drops at the sink. Flow always moves from high to low pressure. so the flow within the phloem moves the sap from the higher pressure sources to the lower pressure sinks. Sugars may be loaded into the sieve tubes by active transport in some plants. This means it requires ATP. In other plants, sugar passively diffuses into the sieve tubes at the source. Diffusion is believed to be the means by which sugars leave the sieve tubes at the sinks, though this is still under study. The water that exited the phloem via osmosis at the sink either stays there or is returned to the xylem. Thus, some water is transported via the phloem, but the volume is small compared to the movement of water through the xylem. Where phloem sap ends up is determined by the sinks. Phloem transport to reproductive organs appears to have priority over movement to stems and leaves, and these have priority over movement of the phloem sap to roots. The phloem also supplies carbohydrates to organisms outside of the plant, specifically fungi and bacteria that form symbiotic associations with plant roots. A fraction of the carbohydrates transported to the roots spills out into the rhizosphere, the soil layer that surrounds actively growing roots. This supply of carbohydrates stimulates the growth of soil microbes. As a result, the densities of microbial organisms near roots are much greater than the rest of the soil. These soil bacteria are important decomposers of soil organic material, which is rich in nutrients such as nitrogen and phosphorus. The release of carbohydrates at the root is therefore thought to enhance the ability of roots to acquire nutrients from the soil. And we'll discuss this more in a moment. Plants make a lot of roots. With the exception of sunlight and carbon dioxide, everything a plant needs to build and sustain its bodies enters through the roots. This includes water as well as nutrients that play important roles in metabolism and structure, including nitrogen, phosphorus, and potassium. Young, actively growing roots are the site of the majority of water and nutrient absorption. Roots have an outer layer of epidermal cells like the stems and leaves. Near the tips of growing roots are slender single-cell outgrowths called root hairs. The extended branching of roots in the presence of root hairs creates a large surface area in contact with the soil that is available for nutrient absorption. As we move into the root from the epidermis, there is the cortex. This is part of the ground tissue and is comprised mainly of parenchyma cells. The thickness of the cortex varies from species to species. At the center of the cortex is the vascular tissue, the xylem and the phloem, which is surrounded by a layer of cells called the endodermis. The endodermis functions as a gatekeeper, regulating which nutrients can move into the xylem from the cortex. Let us zoom into this cross-section of the root to focus on how water and ions from the soil move into the root, through the cortex, and then past the endodermis and into the xylem. Epidermal cells, including root hairs, use protein transporters to increase and regulate the uptake of nutrients from the soil and to actively excrete solutes not desired by the plant. Alternatively, water and nutrients can enter through the relatively porous cell walls that surround each cell. Water and nutrients can then travel across the root cortex by either going from cell to cell via plasmodesmata or by traveling through and along the cell walls. However, at the endodermis, flow of water and nutrients through the cell wall is blocked by the Casparian strip, which is a thin band of hydrophobic material that encircles each cell. Water and any dissolved materials can only gain access to the xylem by passing through the cytoplasm of the endodermal cells. The significance of the Casparian strip is that it allows the root to control which materials enter the xylem. By using the energy of ATP to control the movement of materials across the plasma membrane of the endodermal cells, roots can concentrate nutrients that are needed in large amounts and exclude those that can damage plant cells. Roots expend a lot of energy acquiring and absorbing nutrients from the surrounding soil. However, there are soil microorganisms that are much more adept at obtaining certain nutrients from the soil. Many plants have evolved symbiotic relationships with these fungi and bacteria to gain access to these nutrients, in particular nitrogen and phosphorus. Mycorrhizae are symbioses between roots and fungi that enhance nutrient uptake for the plant, and particularly for phosphorus. In return, the fungi receive carbohydrates produced in leaves and transported to the roots by the phloem. There are two main types of mycorrhizal relationships. The first, ectomycorrhizae, produce a thick sheath of fungal cells that surround the root tip and the walls of the root cells. The root and the fungal cells are able to exchange carbon and nutrients through their plasma membranes. The second type are endomycorrhizae. The fungi in this relationship do not form structures that are visible on the outside of the root. Instead, they form highly branched structures called arbuscules that protrude into the interior of root cells. The arbuscules increase the contact area between the fungus and the root cells, enhancing carbon and nutrient exchange. Both types of mycorrhizae produce a network of fungal hyphae that extend out from the roots to obtain nutrients from the soil. Plants also form symbiotic relationships with bacteria. In many ecosystems, nitrogen is the nutrient that is most likely to limit growth, as it is the nutrient that plants must acquire in the greatest abundance from the soil. In of itself, nitrogen is not a rare element. Almost 80% of the atmosphere is nitrogen gas, or N2. Unfortunately, plants and other eukaryotic organisms cannot use nitrogen gas directly. Only certain bacteria and archaeons have the metabolic ability to convert nitrogen gas into a more biologically useful form via the process of nitrogen fixation. Nitrogen-fixing bacteria can transform nitrogen gas into ammonium ions, which can be used to build proteins, nucleic acids, and other compounds. Many nitrogen-fixing bacteria live freely in the soil, and some live in plant roots. Some plants form symbiotic relationships with nitrogen-fixing bacteria when the levels of available soil nitrogen is low. The establishment of this relationship starts with chemical signals transmitted between the roots and bacteria. Bacteria then multiply within the rhizosphere, and root cells begin dividing, leading to the formation of a root nodule. Root hairs or breaks in the epidermis allow the nitrogen-fixing bacteria to enter the nodule, where each bacterium will be contained within a membrane-bound vesicle produced by the host plant cell. The plant supplies its nitrogen-fixing bacteria with carbohydrates transported in by the phloem and carries away the fixed nitrogen in the xylem. Few plants have developed a relationship with nitrogen-fixing bacteria, though the bean family is well known for forming this type of relationship. Those that do participate provide an important way in which biologically available nitrogen can enter the ecosystem. If you have any questions, please let me know via the Q&A discussion board.